Welcome to Likable Science here on ThinkTech Hawaii. I'm your host, Ethan Allen. Joining me in the ThinkTech studios today is Dr. Dave Carter from Chaminade University. Welcome, Dave.
Well, thank you for having me.
It's a pleasure here. We're looking forward to a good conversation today. As we always do in Likable Science, we try to bring out the very accessible and appealing nature of science, and we have some interesting topics today. Dave is a forensic scientist, and so we are going to be talking about the dynamics of death. A lively topic here.
Yeah.
So Dave has a broad background here. He got his Bachelor of Science in archaeology at University of Idaho, did his graduate work in England at Bournemouth University in forensic anthropology, and then finished off with a doctorate in James Cook University in Australia. So you really traveled around the globe.
Yeah, that's right.
You're a well-rounded individual here.
Yeah.
Well, excellent. So I guess my place to start here is, how did you get involved in this? What drew you to studying forensics, to studying how things die and decay and all that? It seems like an obscure area, you know?
Yeah. Well, that's an excellent question. And to be honest with you, I can only answer so much of it because it's something that just kind of presented itself to me, where I'd always had an interest in history, obviously had an interest in the skeleton, and that's why I got into anthropology and archaeology. And I just remember one day during my undergrad education where I appreciated that history isn't always 100 years ago, thousands of years ago, millions of years ago. Sometimes history is earlier today, or last week, or last month. And once those two ideas came together, just the interest of the skeleton and that history can be quite modern, forensic science really presented itself to me, because a lot of what criminal investigators do is very similar to what archaeologists do. So from my perspective, it was a very natural progression.
Yeah. Interesting parallel between criminal investigation and anthropology there. Funny you should make that. Yeah, I was just reading today, there's somebody doing some very interesting work now on the Black Death and looking at the changes in sort of human health brought about by the Black Death, and analyzing skeletons from before and during the Black Death and those afterwards, and finding that it really sort of cleaned out, I guess, sort of called out some weaker type people.
Absolutely.
And the people who came afterwards were living longer, healthier lives.
Absolutely, yeah. It had a huge effect on civilization, that disease.
Excellent. So what kinds of backgrounds should a student sort of study or get into to if they wanted to pursue this kind of field? Is it really a narrow course of study, or can they jump into it from a broad range of topics?
No, it's relatively broad. What's going on nowadays is employers are more looking for people with a natural science background. And it doesn't have to be a degree in forensic sciences. It can be a degree in biology, biochemistry, chemistry. Those are probably the four most common, including forensic sciences. This has been somewhat of a shift since really the 90s, where more investigators would have backgrounds in the behavioral sciences, political science even, psychology, anthropology, sociology, whereas the lab scientists would be more of the natural science background. And now we're seeing more of the investigators having their degrees in biology and chemistry, because so much of forensic science is getting out into the field now.
Right, because it's a fairly broad field in the sense it covers very specific medical issues, it covers criminal issues.
Yeah, it covers all kinds of things that would never make a successful TV show. Termite inspections, food quality, soil quality. I mean, those are all areas of forensic science. Seems like we mostly just latch onto the criminal stuff, though.
So say a little more, if you would, about that termite inspection. What?
Oh yeah. Well, forensic science, the most simple definition of it is just any science used to address a legal issue or a regulatory issue. So if you're dealing with something that is part of the justice system or some quality regulation that's out there, that's technically going to be a forensic science. There's limits out there to what we can allow be exposed to society.
Sure, absolutely. So what does your work day look like? You know, what is a typical day for you?
Yeah.
Or is there such a thing?
Okay, well, you know, first and foremost, my occupation is that of a professor. So I spend most of my time on a university campus. I work at Chaminade University of Honolulu. I have a brilliant administration, very keen students, and so that makes my day relatively unpredictable. I have set teaching times, but when I'm not in class, I might be working on manuscripts or conducting experiments or taking phone calls from lawyers or investigators, people at the medical examiner's office. So there's a lot of unpredictability to it. And sometimes that's stressful and sometimes it's very exciting.
So you mentioned experiments. So can you tell us a little more about what kinds of experiments you do?
Yeah, really the field that most of my interest lies in is a field that would probably be referred to as post-mortem microbiology. And it's a field that's been around for a very long time, over 100 years. So the experiments we conduct are those that are investigating the microorganisms that live on and in decomposing corpses, whether they're human, pig, mouse, rat, any mammal, basically.
So I think we have a video, if I'm not mistaken, that shows one of your experiments.
Yeah, that's right. Yeah, we do.
Maybe we can show that here.
Most of the time I call them maggots. And I really just wanted to capture these videos to show people that a dead body in an ecosystem really serves as it's like an island. It's a hot spot of diversity and life and nutrients and energy, and it attracts a lot of organisms that come to it because those organisms that can use a dead body as a resource, that's an excellent meal for them.
Right.
You know, they don't have the luxury of shopping for food like we do. You know, they have to go out and fight for their meals.
No, that I thought that video there showed how very dynamic that process really is.
Oh yeah.
There's a lot going on there. There's all kinds of different organisms working there, doing their shopping, as it were.
Yeah, absolutely. Yeah.
And fairly rapidly going to pull the whole thing apart. Excellent. That's very very. Some people might say gory, but interesting,
Sure.
certainly.
Yeah.
And so, what sorts of tools beyond? I mean, that looked like you're watching a dead animal decay there, but
Yeah.
you also use, I know, a whole variety of tools, from some relatively simple, commonplace to probably some quite sophisticated tools in your work and techniques.
That's right. You know, going back to the video, the one tool I use constantly is a camera. So if you're interested in forensic sciences, get a decent camera because you're going to take a lot of images because you'll have to document a lot of stuff. In addition to my camera, when it comes to collecting data, one thing I always have with me is some type of data logger. This particular one measures temperature and relative humidity. And I think we'll talk about some of those things a bit later. Those are environmental factors that play a major role in the rate of decomposition. So it's important for us to measure those. Otherwise, we use a lot of tools that are very similar to how an investigator would collect human DNA. We use a lot of sterile cotton swabs. These are how we would collect our microbial samples. And I could just do like a demonstration here real quick. I just take it and rub it on a surface. I can rub it on my hand. And because we're usually out in the field, we want to transport this so we don't get any other microbes on it. So we tend to just put that in a sterile swab. And usually the swab manufacturers and the tube manufacturers should get together because the swab, you know, they never fit. You always have to break them off. So just kind of put that in there, get it back to the lab. And then the thing that's most interesting for me is just looking at microorganisms and taking images of them and watching them grow because there's such an microbes are such an interesting component of the tree of life. You know, it's interesting to think of life from their perspective because it's so different from human life.
Right.
But we get back to the lab, we take our swab out, and then we can just take a Petri dish and, you know, we just rub our swab right on it and put the lid on, and then we just let it incubate, usually about room temperature. And I'd say certainly in 48 hours we'll have bacterial cells visible to the naked eye on there, possibly in 24 hours.
And you can tell because of the different types of bacteria or other microorganisms, gives you clues about what the tissue that you were doing was, how long it had been lying there, and a variety of different things.
Yeah, we're really starting to get some interesting insight now, just within the last 5 years. And if there's any viewers out there that want to look into the Human Microbiome Project, Earth Microbiome Project, American Gut, a lot of what we're finding is parallel to those studies that are taking place. We're learning that as a body decomposes, the structure of those microbial communities change over time, which makes a lot of sense because their resource is changing. We're also learning that that change is predictable, meaning that if we have certain parameters, we can take a swab of a microbial community, plug it into a regression equation, and it will give us an estimate of how long that mammal has been dead. We're also finding that environments that people spend a lot of their time exposed to, like where we live, certainly a dwelling, a place of work, the surfaces in those areas take on a microbial community that's similar to the people that are around them. So it looks like there's going to be a lot of potential in the next few years for using microorganisms to trace individuals to particular places and objects.
So it's not just you're shedding your DNA around your house, you're actually shedding your microbes that are, by looking at those populations, they can say, this was Dave was here rather than Ethan was here.
That's right, and you're probably shedding about 10 times more microbial cells than skin cells.
Right, because you have about 10 times more microbial cells even on you than you do your own cells, right?
This field of forensic microbiology is, it's a real blessing to be working in it right now because it's incredibly exciting.
No, it seems to me that should be much more complex though, because you're dealing now with a whole slew of species and you've got to tease out what genetic material belongs to what species and then be able to sort of reconstruct that population and then compare that population back to the individuals.
That's right. Yeah, it's highly complex. A lot of computing power is required. Even just the simple culturing of microbes is complex because there's so many assumptions being made. You know, we can only culture so many microbes. So we know right off the bat we're not getting them all. So then we sequence profiles to get them all. Well, then we get these massive data sets where you have 3,000 microbial taxa that you're trying to wrap your head around. And it is highly complex, and it can take a lot out of your brain when you're working on these data sets for a while.
I can well imagine. And it must be, I mean, because there's so many different factors. That is, you take a given sets of populations of microbes off of your skin, you put them on a petri dish, and some of the ones that were on your skin are not going to do well there, and others are going to do extremely well. And so the ratios of those populations all radically change.
Absolutely.
And you have to take this into account, presumably.
That's right. Yeah. So you could even call it, there's just a lot of educated guessing taking place, you know. Okay. It's really interesting.
Yeah. And then, of course, yes, all that, as you say, changes over time with the decay processes, again, feeding certain populations, crashing other ones.
Yeah.
Yeah. Huh. Interesting. So, do you get involved in things then like pulling out the DNA and amplifying that, or do you leave that to other people, or
No, I collect samples. When it comes to the molecular side, my responsibilities are to collect samples and then provide them. The sequencing is done either with my collaborators in the Knight Laboratory at the University of Colorado Boulder, or we also work with a company in the Bay Area called Second Genome in San Bruno, California. So my job is to design experiments, collect samples, send them off for sequencing, and then interpret the data sets that come back. So in spirit, what my job would be very similar to a crime scene investigator, where they would go collect evidence. Some of the evidence they would send to a crime laboratory. The crime lab scientist would process it and provide a report back, and then it'd be the investigator's job to bring everything together and make a story.
Huh, intriguing. So there's a lot of, it is very highly collaborative then.
Yeah, it has to be. Yeah, it has to be.
In this day and age, you can't sort of do it all yourself.
No, no.
Yeah, Sherlock Holmes is sort of almost a thing of the past in a way, right?
In some ways, yeah. In some ways, yeah.
Although from Conan Doyle's work, I mean, Holmes was virtually the computer in and of himself, right? I mean,
yeah.
He had all.
I mean, Conan Doyle was ahead of his time.
Yeah.
Agatha Christie even had some elements ahead of her time, or certainly contemporary with what forensic science was doing then. So, yeah, really neat stuff.
That's most interesting. So, do you ever get involved in active sort of criminal matters?
Sure.
And can you what does that look like? I mean,
yeah, that's it's certainly not a day-to-day component of my responsibility. I contribute something to, I'd say, a couple dozen cases a year, and sometimes it's as simple as I get a phone call from an investigator or a lawyer or somebody that wants some information, and I can provide them with a name of somebody they can talk to. So sometimes I'm really just serving as a middle person because I have a network of expertise that I can help people out in. In the cases where my expertise is desired, those are death investigations, usually an investigation where the amount of time that's passed since death is uncertain. So I try and help investigators figure out how long people have been dead. Sometimes I'll help investigators try and find bodies that were meant to be hidden. An investigator will have a lot of evidence that leads them to believe that somebody is dead, but they don't have the body yet. So sometimes we have to try and help find bodies, and it's very difficult to do.
And how do you go about doing that?
Well, there's a lot of ways. In those cases, a lot of times you're dealing with something that's outdoors. And that's what makes it so difficult, because a body, imagine trying to find one body even on the island of Oahu. You know, a lot of people say this island's very small, but if you said, okay, go find one body, I've hidden it somewhere on the island, it'd be a very tough job. You can look for changes in vegetation. You can look for insect activity that might, if you see a lot of insect activity, you might simply just follow flies to where the bodies are, looking for bones, looking for any changes in the environment that takes place, either from humans going into the environment to dispose of a body, or the body itself affecting the environment as it's decomposing. A lot of times a body will kill vegetation at first, but then about a year later the vegetation comes back very lush and very green.
It's fertilized.
Yeah, it's a massive, And so you can use little changes like that to try and find things.
Interesting. Actually, it's funny, it ties back to a show I was doing recently on systems thinking, and this is all, you think of the body lying there more or less passively and decaying, but what you're saying is there's actually a very active interplay between the organisms around it, the organisms within it, the tissues decaying, the organisms that are growing there,
and we really don't know much about it, but like you say, it's highly interactive, complex, and it's because it has to be. You know, we live in a universe that energy can't be created or destroyed, so organisms must decompose. You know, leaves must fall from trees and break down, otherwise all of our energy and nutrients would be locked up in these living things. So it's so attractive and so dynamic because it's required for our systems to function.
Right. One of my coworkers says, what is it? Energy flows, matter cycles, life networks.
Right. Okay. Yeah.
So that's most apropos to this discussion. So that's a very interesting, novel way to look at it. I really had not considered sort of the two-way flow there in between a dead organism and the environment, and how it's shaping the environment as the environment is shaping or reshaping yet.
Right, yeah.
That's quite interesting. I think we're going to take a little break here, and then we'll come back and we'll get into the more of the meat of your topic. I'm Ethan Allen, your host on Likable Science. With me today in the ThinkTech studios is Dr. Dave Carter. We're talking about the dynamics of death.
Hello, I'm Martin Despang, and I'm the host together with the one and only Ali Yamashita, and our show is called Urban Transcendence. And Urban Transcendence is about identifying where we have a unique situation of a vibrant city in one of the most beautiful natural environments. So how these two can coincide, sometimes conflict, how they could find reciprocity in the 21st century is what we're excited about. And we're planning on bringing in a diverse body of guests, both from the arts and the science and the established and the wise and the emerging generation. So hope you will join us. We'll always be on Thursdays from 1 to 2 p.m. Thank you.
And you're back on Likable Science with your host, me, Ethan Allen, ThinkTech Hawaii here. With me today in the ThinkTech studios is Dr. Dave Carter from Chaminade University, a forensic scientist, and we are talking about the dynamics of death. So Dave was just telling us about how intriguing interactions between dead organisms and their environments, and how there's a lot of interesting flow back and forth. So let's dig into that a little more deeply now, if we might here, Dave.
Sure.
So what are these typical processes that go on, sort of that we call decay, and we sort of use that one word. It's
right.
Obviously a very complex set of interactions.
Yeah, that's right. I start by thinking of it as two primary processes that take place. Shortly after death, the first process that kicks in is called one that's called autolysis. And that's really just the abiotic breakdown of cells in our body, because our body's not functioning anymore. Our immune system stopped, we're losing oxygen, and cells are losing their integrity and bursting. So we have that process taking place. And again, there's not organisms associated with that, it's just the body losing its ability to function. The other major process that contributes to most of the visual changes we see on a corpse is a process called putrefaction. And you can think of that as being associated with a lot of fermentation. The bacteria that take up space in our gut during life, after death, the rest of the body becomes a potential habitat for them. Oxygen levels are dropping, like I said, immune system's gone. So now they can begin to migrate throughout other parts in the body that they never could before. As they're metabolizing and decomposing our body, and that's the putrefaction process, they release gases, just like you and I do as we respire. Fluids are being released from their cells, going into places they normally don't go in the body, so you have color changes, bloating of the body. Those processes, from what we're starting to understand now, really help to bring in insects and other scavengers. And when we say, usually we don't call insects scavengers. Usually scavengers are mammals, birds, or reptiles. It looks as though some of the compounds that microbes are releasing as they're decomposing attract insects to the body and attract some of the scavengers to the body. So once those organisms come into play, you then really have a third component, a third decomposition process that technically is called comminution, and it's really the physical aspect of decomposition. So for a scavenger, it could be clawing and chewing. A maggot has some hooked mouth parts that it can use to physically break up a resource.
So should we look at this series of photos that
Yeah, I think we should. Yeah, I think we should.
Let's see. Actually, our first one is really not about that. The first one is actually your workplace, right?
Yeah, this is actually a view from the site where we decompose swine carcasses on campus, and we'll look at a few of those. I like to show this because it's such a beautiful place, but two, it reminds me that what we're doing is just we're investigating one small component of a very large ecological system, and that's just a picture of an ecological system right there.
But then the next picture here.
Yeah. Okay, so there's going to be a series of images coming up now, and each image is going to be approximately 24 hours older than the previous one, just to give you a rate of flow here. This picture was taken of a swine carcass, and it's approximately 4 hours post-mortem. So this pig was killed, driven to campus and placed at our facility here. And you can see it's on a frame-shaped thing. That's really just a basic hanging balance that allows us to measure how much mass the carcass is losing over time. So we would call this a fresh carcass. And after about a day here, these were placed in February on Oahu. You get the bloating that I talked about. You can see the shape of the carcass is changing. That bloating's the result of putrefaction. You know, those microbes inside are decomposing and releasing gases. The other process you can spot if you look at the limbs and when they look stiff is a process that most people probably heard of called rigor mortis. And that's the temporary stiffening of muscles and joints in the body. If you've ever driven down the road and seen roadkill on the side, if you catch it at the right time, you'll spot rigor mortis, but it goes away after a while. The next image here, this is about another 24 hours after that. This is a really interesting picture, actually, because there's a lot of potential investigative information right in front of our face here. One, you can see the abdomen has ruptured open. Okay. First thing you need to know is that doesn't always happen. In my experience, that's not common. I'd say it happens in 2 out of every 10 carcasses. But I think that's a reflection of the regions I've been working in, because I know other forensic scientists where rupturing is actually a very common process that they see. When I see rupturing, it's because of one of two things. Either a scavenger has come along and started to scratch at the corpse, and that scratching, along with the internal pressure from the bloating, just collapses the skin and it bursts. The other time I've seen it happen is when you have a corpse or a carcass that spends most of the day in direct sunlight, and you're in a very warm time of the year, July, August, and what happens there is microbial activity is so great that the microbes are releasing gases so fast that they can't come out of the natural orifices, and so the body just pops like a balloon. Now, one thing I want to point out here is everybody should look at the head of this carcass. Now, in my experience, what happens the 80% of the time that rupturing does not happen is usually you get a nice clean skull to start off with, because that's where flies lay most of their eggs. They like the eyes, the nose, the mouth, the ears. It's a perfect nursery for maggots, you know? So since that's the first place maggots start feeding, it's the first place that usually becomes clean, unless you have a significant trauma to the torso. So from an investigative point of view, if you were to come to a body during investigation and you saw a skull that still had a lot of soft tissue on it, you should be open to the possibility that their torso experienced significant trauma. Maybe multiple stab wounds, multiple gunshot wounds, car accident, something like that.
Things that would provide homes for the maggots,
basically. Yeah, they're basically distracting the maggots, where they'd normally say eyes, nose, mouth. Now they're saying, well, why don't we just go straight to the guts? You know, so just a little trick you could use as a death investigator there.
Excellent. So 24 hours later here?
Yeah, and really this image is just here to show you that carcasses don't last long. Here on Oahu, even in the coldest time of year, you're going to get to a stage of decomposition like this in about 7 days, 10 days. So if you're searching for a body, you don't have a lot of time to find a body, certainly that's still recognizable to identify. But after just a week or two, you're going to have bones, teeth, hair, dried out skin. There's not going to be much to work with. And that's what we're trying to help.
Interesting how varied nature is, because in some environments you can get incredible preservation of a lot of tissue for incredible lengths of time, right?
Yes, you can. Previously I was at a university on the mainland, the University of Nebraska, where you have very clear seasons,
right.
very hot summer, very cold winter. And one aspect of these studies that has been done relatively little is looking at seasonality, actually. Most people decompose corpses and study them in the summertime. Very few people have put them out in the winter, so that was one thing we did. And you're exactly right. We found that if you time it right, you're essentially putting your carcass in a freezer.
Right.
And we would go out 60 days after death, and it looked like that pig just died. It was really interesting. Other places that are very dry and very hot, you can get natural mummification. Or other places that are very dry and very cold, you can get preservation.
Even some swamp areas, I guess, because of tannins and things in swamps will essentially preserve in a different mechanism.
Yeah, that's right. Yeah, anaerobic, bogs. Yeah, a lot of tannins, a lot of things that keep biological activity down. Yeah.
Intriguing, intriguing. So this is then, these processes are really run by a whole set of organisms. It's not just the internal organisms, and it's not just the flies that come in afterwards, right? I mean, it's a sort of a ballet, as you might say, right?
Yeah, it's becoming to look like a very complicated food web, where right off the bat we're starting with a lot of microorganisms, because every mammal has a lot of microorganisms on them to begin with. And then we're bringing in insects, sometimes vertebrate scavengers, but then there's a whole bunch of other microorganisms that are playing a role in decomposition. It's not just the bacteria that are mostly what's taking, those are the most active in the body, but you can have fungi, nematodes, protozoa, slime molds we're seeing. So there's a lot of organisms out there that take advantage of this resource.
Well, yeah, as you say, it's essentially a rich food source for a large, a whole ecosystem in and of itself, basically.
Yeah, absolutely, yeah.
It's like in the ocean that you're now finding whale carcasses that in the middle of sort of a more or less a desert on the deep ocean floor. A whale carcass will support huge colonies of all kinds of organisms for apparently quite extended periods of time.
Yeah.
So very slowly decays, of course.
Very similar process. And I try and keep up with the whale fall stuff too, because there are a lot of similarities where we're starting to recognize carcasses as, it's a resource that promotes biodiversity, because there's some organisms that need a carcass to reproduce. Or it serves as a corridor for organisms to move within a system or even between ecological systems. So in terms of, if you're interested in ecology, the role of biodiversity and the function of an ecosystem, a carcass serves as a really nice model to investigate some of those aspects.
Sure, well, it's one of, in very sort of fundamental ecological terms, there are producers, there are consumers, and there's decomposers,
That's
right?
right. That's right.
You get nowhere without that third part of that
That's right.
triad, right?
That's right.
They can't all just flow one way.
Yeah.
Well, that's super. So, and I think we'll get into the. Were there more photos on this decomposition set? I'm not sure.
Yeah, I put one image here just to end, because this is at the same decomposition site, but this is a different carcass. I put this one on here just to kind of reinforce a bit what we were talking about with the skull becoming exposed early on. You can see, even though this carcass also doesn't have a perfectly clean skull, a lot more of that facial soft tissue is gone. And really the point of this image is just to show you that there's a relative amount of variability when a carcass decomposes, because it's such a highly attractive resource. That's why I say a lot of times, in my experience, most of the time, because there's no one way a carcass breaks down. There's a lot of different ways it can break down.
Sure, no hard and fast rules. Well, excellent. I'll tell you what, we're going to get into the more microbial level here when we come back.
Excellent.
But we're going to take a little break now. You're with us on Likable Science here, talking about death and decomposition here with Dave Carter from Chaminade University. We'll be right back.
I'm Jay Fidel. That's Sharon Moriwaki of the Hawaii Energy Policy Forum. And every Wednesday we have Hawaii, the state of clean energy. We've been doing it for some time now, and we have some fantastic guests on there, unbelievable guests who give us insight into what is going on in a very complex, sometimes very confusing, sometimes very disappointing area of progress in the state. So we love doing this. We love meeting them. We love talking to them. We love having their ideas out on the table. So maybe, just maybe, we can all make some sense of what's going on. Sharon, what do you think?
I think that's absolutely correct. We enjoy ourselves meeting with all these people and hearing about the energy and the state of clean energy, and hopefully we advance clean energy for the state. So it's terrific. Join us.
Okay, it's every Wednesday. Okay, Wednesday is Energy Day. Every Wednesday, 4 to 5 p.m. Hawaii, the state of clean energy, here on ThinkTech Hawaii. We'll see you there.
And you're back on Likable Science. I'm your host, Ethan Allen. With me today in the ThinkTech studios is Dr. Dave Carter from Chaminade University. We're talking about forensic science, the dynamics of death, decomposition, and all kinds of interesting stuff. So, you've mentioned now, Dave, several times that the bacteria play a critical role, and that these are an interest of yours.
Yeah.
And I think we have some photos of some of these are, I guess, from some experiments you've done.
That's right.
Of some of the petri dishes and all. We might sort of start with one of these and move into that.
Yeah, this is a good one to start with here. Like I demonstrated a few segments back, first thing we do is just rub a sterile swab on something we want to collect a sample from. And then when we get into the lab, we rub it on a petri dish. And these are what the petri dishes look like. If you look at those streaked patterns in there, you'll see there's a bunch of different colors, a bunch of different shapes of bacterial colonies. So this image, I just wanted to show you all the variability we have in bacteria we're getting off these carcasses. And what our next step after this is to try and isolate what we see, what we think are different species. You know, here's another example of a petri dish with multiple microbial taxa on it, because that's the environment. You know, there's all kinds of microbes out there, and when we bring them into the lab, we're bringing all of them in and culturing what we can. And so sometimes we have to go into something like this that's kind of soupy or slimy or whatever you want to call it, and try and isolate different species out.
Uh-huh. And do you use always pretty much the same medium to grow, or do you do multiple sort of streams of media to pick up different things?
That's an excellent question. There's a bunch of different media that we'll use, but when we're first trying to understand a particular process or a particular environment, like we're doing with our decomposition site here, since I've only been here 18 months, still relatively new to me, I usually just stick to one growth medium. And all these images are with that one medium. It's just a standard nutrient agar. It has beef extract in it and other nutrients. And it's meant to just be a general microbial food source. But I think, as you and I were talking about off camera, we're only growing a small percentage of what's actually there.
Sure.
So that's one thing we always have to take in consideration when we culture microbial taxas. We can't culture everything.
Right. So this next plate here showing?
Yeah, and this is showing what an isolated petri dish can look like. So we've gone into one of those dishes that has multiple microbial taxa and tried to pull out what we think is just a single microbial species and then spread that on its own petri dish. And so this one here is something from the skin. We haven't identified this one yet, but it's probably a gram-positive rod-shaped bacterium, just because gram-positive rods, gram-positive bacteria in general are very common on skin anyway.
As opposed to this next image that
Yeah, well, this is the interesting thing, because this is probably a gram-positive bacterium as well, but this one is probably like a micrococcus, a gram-positive coccus, which is more of a round-shaped microorganism, round-shaped bacterium, whereas the rods are more shaped like a pill you take for medicine, or if you follow the Despicable Me films, they're shaped like the Minions in Despicable Me. So our goal is to isolate these things based on how different they look, and you can see this one looks completely different to the colonies that we saw before. And then we identify them and get as close to species as we can. Sometimes we can't always get to species.
Right. I guess the whole species thing with bacteria is actually a pretty iffy game in some sense anyhow. Bacteria do a lot of swapping of genes and...
Oh yeah. You know, and microbiologists can use a slightly different taxonomy system. Like, for example, you probably come up through biology being taught about kingdom, phylum, class, all that. I don't use kingdom at all. I use domain, phylum. So there's a lot of conversations going on about that right now. It can be kind of confusing.
Interesting. So your next Petri dish here looks a little different.
Yeah, this is more, I wanted to bring an image like this because this is more of your stereotypical isolated Petri dish, what it would look like. A lot of times it would even have an additional line going on towards making a pentagon shape there. This is just an isolation process, and what the point of it is to try and get individual colonies growing on their own, and you can see that up at the top of the Petri dish there. You have distinct, relatively round-shaped colonies up there, and you want to do that just because it's easier to go in and pick them out and just isolate those.
Well, that's intriguing, and so you can do all these techniques on these bacteria. Then you can look at their genetic material and amplify that and tease different groups apart.
Yeah, that's right. Where most of my thoughts and work is at the community level. So I'm looking at a collection of different species, trying to figure out what they're doing, what roles they're playing, and how they change over time. But we're also, every species we bring in and identify, we also put in a long-term storage collection on campus, because I can anticipate we're going to be running across a lot of microbes that we still need to do work on their genome. And so if anybody wants access to these bacteria that we're preserving, we can provide access to those, and people can look and go through and get all the details of their genome out.
Excellent, excellent. So where is this all sort of headed now? What's the next big thing coming up in forensic science or in the aspects particularly that you're studying?
Sure. Well, I'd say, obviously I'm biased, but forensic microbiology is probably one of the hottest areas of forensic science right now. And a lot of that's just because we have science and technology that we didn't have 10 years ago. You know, now we're able to go to locations, swab objects, swab dead bodies, and identify microbes that we never could identify before. And that application of microbiology to death investigation is one of the emerging areas in forensic science right now. I can also see the emergence of using microorganisms as trace evidence, so to associate a particular person to a place or an object, trying to match the microbes from their skin to the microbes on an object that they're either often in association with or often touching, like the back of a cell phone or a keyboard or a computer mouse. Just in general terms, I really see forensic science getting as much science into the field as possible. We're getting all these investigators now that have backgrounds in natural science. So they understand genes and genetics and organic chemistry, physics and mathematics. And we're getting the technology that now you can take into the field with you and do things we never could do before. I mean, even just a smartphone nowadays is incredible.
Right.
You know, I mean, there was nothing like that even when I was a grad student, you know? And so I think now we're going to be looking at any microscopy that's portable, any chemistry that's portable, any biology that's portable. I think what we're trying to do now is get as much science into the field as possible so that the crime laboratories will not only get probably more evidence sent to them, because we have scientists as investigators now, the evidence that will be sent to them is probably going to be better, because, you know, an investigator's not going to send what they think is a human hair to a crime lab and say, hey, can you tell me this is human? Now we're getting investigators, they can tell you if it's human hair or not. They need the crime lab to say, hey, can you get any DNA out of that? You know, so it's a very exciting period for forensic science right now.
Yeah, you're getting more and more information from less and less,
Yeah.
smaller and smaller samples.
No
now.
question, no
Yeah.
question.
And interesting what you were just saying about taking like microscopes into the field. I had Manu Prakash
Yes.
on the show a while ago, and he has this, his pocket-sized 2,000 power, very rugged scope, and one can envision this playing a role then, where an investigator could very easily carry a very powerful optical instrument.
Yeah, and that's a perfect example, and I think that's how it should be. Because you can imagine back in the beginnings of forensic science, that type of development would have been equivalent to the camera. Well, let's capture images, and now we have this permanent document of a scene. Now, if we can take microscopes out, it's a very similar thing.
Yeah. That's amazing how the explosion of information, though. So you guys, to some extent, must be sort of similarly overwhelmed in that you're getting masses and masses and masses of data clearly from the videos you showed. Obviously, you have a lot of things there, a lot of stuff going on, and so you might potentially get, as you say, thousands of species involved.
Oh yeah. I'm actually quite tired right now, to be honest with you. But yeah, you're right. Supercomputers are involved with processing these data. Robots in the laboratory setting are used to help streamline the actual chemistry process. We're doing things with microculture now, where culturing microbes just on a micro scale. That's probably one of the most difficult aspects of it, is wrapping one's head around these data sets, so that the scientist's job is ultimately to say, hey, this is what all this means.
Right.
It's very hard to do that.
Oh, yeah.
Just because, like I say, you'll have thousands of microbial taxa, and what are they all doing? We don't know that yet. Maybe the most important one is actually one of the least abundant. We don't know. We don't know.
Yeah, and that could give you very critical information if there is some perhaps not abundant one, but that's associated with a very specific environment or a very specific kind of situation. That might be a critical piece of the information there.
That's right.
It's very easy to lose track of that in the middle of being swamped out with the multiple others.
Yeah, it is.
Interesting. And so what advice would you give to students coming on into this field? How would you tell them to prepare themselves?
Yeah, if you like science, the natural sciences, I think take as much science and math as you can. In high school, going into an undergraduate degree, I would encourage them to invest in patience. There's a lot of people, as a scientist in general, but also as a forensic scientist, and that's for many reasons. Forensic science is quite popular right now, so the jobs out there are very competitive. You have a lot of people applying for relatively few jobs. And the job process is a long one, because oftentimes it involves a background investigation. A lot of those employment series include an examination. So an employment process might take 6 months. So one thing I advise our students to do at Chaminade is start applying for jobs. If you graduate in May, the January. If you graduate in December, the August. The start of your last semester, because sometimes it takes a long time. Contact places that you'd be interested in working and ask them what they want in an employee. Do you want people with a chemistry degree? Do you want people with a forensic sciences degree? And find out, and then do that. And just to make yourself as attractive as possible. And probably the best advice I can give for somebody that wants to work in the forensic sciences is, if possible, do an internship at the agency you'd like to be employed with. It gives them an opportunity to, it's really kind of like a long-term interview, but it gives you exposure to how they operate. You get to see what some of the day-to-day aspects are. And you'll find a lot of agencies end up hiring good interns that they've had because they're already somewhat trained.
Sure. It's sort of like the old apprenticeship model in a sense.
Absolutely.
It's the great way to know whether, again, sort of all the aspects of the ecosystem fit, whether your knowledge, your background, your temperament fits with the company organizational ambiance and needs and ways of functioning, right?
That's right.
Yeah. So they all fit or they don't fit, right? You either thrive in that environment or you don't thrive, just as the organisms that you're looking at there.
Exactly.
Some of them thrive and some don't.
Exactly.
Excellent. Well, this has been a most enjoyable chat with you here. I feel like I've learned a whole bunch, and I hope maybe we've inspired some future forensic scientists here.
Yeah, well, it's been a real honor. Thank you very much.
My pleasure entirely. You've been watching Likable Science with Ethan Allen. I'm your host, and today Dr. Dave Carter from Chaminade University has been with me. We've been talking about dynamic death. Thank you, and I hope to see you again. Stay tuned.